In this video, we're going to talk about post-transcriptional processing of the mRNAs. Now, for prokaryotes, you don't really need to process your mRNA. In fact, the protein synthesis can still happen while you're still making your RNA. That's not true of eukaryotes. So, from RNAP2, the RNA we get out, it's called the primary transcript. However, these are usually not functional and they have to be altered before they are functional. And we have three major types of post transcriptional processing that we can do. One is exo and nucleolytic removal of the polynucleotide segments. Another one is modifying the ends of our RNA. And the last one is modification of specific residues. And the different types of RNAs are all altered differently in prokaryotes and eukaryotes. And so we're going to talk about eukaryotes here. And the 5' end of mRNA have a cap. And this cap is 7-methylguanosine as shown here. And this will be added before the mRNA is done. It will be added when the mRNA is roughly 30 nucleotides long. And there's a couple enzymatic reactions that are responsible for this. First, you have to remove the phosphate from the 5' group. Then you have to add a guanine, so guanylation, by the enzyme guanyl transferase. Then you have the methyl, because it's 7-methyl, and this is done by guanine 7-methyl transferase. Now you can also modify this cap further. It could have a oxygen at the 2' methyl group at the 2' oxygen, so O2' methylation. And this can have at the first and second nucleotides as shown here. And this capping enzyme, what's interesting, and we'll see this kind of like a trend, is that the capping enzyme binds to RNAP2's phosphorus CTD, C-terminus domain. And so we talked about how the CTD is important. This is another way it's important. Now, this cap is specific to mRNAs only. Other RNAs aren't capped. And this 5' cap will be bound by another complex called the cap binding complex. And this complex is also interacting with the phosphorylated CTD. And this capping has a purpose. It's allowing the mRNA not to be destroyed by 5' exonucleotic degrading. So it can't be destroyed from the 5' end when it has this cap. Now, along with the cap, mRNA also has a poly A tail. And it's in a lot of eukaryotic mRNAs, roughly 250 type nucleotides long, shorter in yeast. And the reason for this tail is that during transcription, release of mRNA doesn't always happen at the same point. It's actually kind of hard to do. So if you didn't have a tail, all your mRNAs would be different lengths, and you would make proteins that had different amino acids at the C terminus. And you don't want that. So what happens is that for mRNA, transcription will be cleaved, the transcript will be cleaved from 10 to 20 nucleotides downstream from a highly conserved AAU AAA sequence. So all primary mRNAs will have this sequence. And there will also be a U-rich or a G plus U-rich sequence. And the cut will be less than 50 nucleotides away from this. After this cut's made, we can make this poly A tail by the protein poly A polymerase, or PAP. And it doesn't matter what RNA polymerase that you use, you're going to add this cap to your mRNA. This PAP is activated by another protein called the cleavage in polyadenylation specificity factor, or CPSF. Now, CPSF, like the previous proteins we talked about, can bind to RNAP2's phosphorylated C terminus domain. Now what's interesting is that you actually don't need poly A for this mRNA to be translated. And what happens is that this tail will be shortened as it ages in the cytosol, which is suggesting that this tail is protecting the mRNA from being destroyed by the 3' end. And in the cytosol, these poly A tails actually are binding another protein called the poly A binding protein, or PABP. Again, this protein is preventing your mRNA from being destroyed. So the 5' and the 3' regions are really designed to make sure we don't prematurely destroy our mRNA. And that's it for this video. I will see you in the next one.